Some of the sequencing techniques are the ones which I listed here. There are so some of the sequencing techniques are here: whole genome, whole exome. So basically, if you classify the genetic material broadly, one is DNA and the other is RNA. So understanding the DNA, we have various techniques. So we choose the technique based on the research question we are trying to address. So for DNA, the commonly used techniques are whole genome sequencing, whole exome sequencing, targeted sequencing. So all these genome, exome, and targeted sequencing is to understand certain parts of our genetic or DNA material. So when I say whole genome sequencing, it is trying to understand the composition of our entire genetic material present in our cells. Whereas whole exome sequencing is just to understand the coding part of our DNA, and targeted sequencing is see like if I say like our entire genetic material is made up of around, I would say, like 23,000 to 25,000 genes. So if our interest is a set of 10 genes, then we go with targeted sequencing. So that is how we pick the sequencing technique according to our interest. And then the other techniques for DNA study is ChIP-seq, ATAC-seq, and bisulfite sequencing. So those are majorly used in epigenomics, where we try to understand the methylation patterns of DNA. So there are these kind of methylation patterns which regulate downstream processes in our cells. So it is these techniques are used to understand the methylation patterns of DNA. And then the RNA sequencing is called transcriptome sequencing. So when if you want to understand the composition of our entire RNA, we call it whole transcriptome sequencing. And if it is only a specific region of RNA, we call it targeted RNA sequencing. And then microbiome sequencing or metagenomics, where we try to understand the genomic sequences of bacteria, fungi, viruses, and all kinds of microbiomes. We call it metagenomic sequencing, and the sequencing techniques are 16S rRNA and 18S RNA sequencing. And then there is this advanced sequencing technique called single cell sequencing, where we sequence the genetic material at at a single cell resolution. So this is just to give an overview of what are all the sequencing techniques available uh, for understanding the DNA or RNA in our cells. So now there are so many techniques. So what kind of information or data can we expect from these techniques? So I mean, like we apply each of these techniques for a purpose to solve certain kind of research questions, and there is certain kind of information coming out of all of these analysis techniques or sequencing techniques. So, for example, DNA whole exome or whole genome sequencing, majorly we look at the genetic changes present in the sample of our interest. So we compare the sample of our interest with the reference genome, so which is generated in our human in our case it is the reference genome came out of human genome project, where now we have the composition of our human genome and the composition of human genome present in the normal individuals. So now when I'm analyzing a sample of a disordered sample. So then I compare the genome of that with the reference genome, and then the prominent ones which we look at in the whole genome or exome sequencing is point mutations. So these are different kinds of mutations which are listed here, like point mutations, indels, copy number changes. So it is like wherever the genetic changes are seen are all reported from whole genome or whole exome sequencing. And transcriptome sequencing is like sequencing the RNA, where this is done at a certain conditions. If you want to compare a different treatment condition, say how the treatment work, I mean like how the genes are expressing before the treatment and after the treatment. Is there any kind of improvement, or what are the set of genes which are involved in a disorder, or those kind of information we apply transcriptomics or RNA sequencing. And then, like I already mentioned, epigenomics is like to understand the methylation patterns, and to also understand the protein DNA interactions. We use this ChIP-seq, and ATAC-seq is for understanding the regulatory regions of our DNA. So, and then the latest hot topic in NGS is the metagenomics, where we try to understand the composition of microbiomes. So, given a soil sample or given a gut microbiome sample, like where we have a diverse set of microbiomes present in those samples, right? So, to understand that, and the other another important or interesting application is this antibiotic resistance. So these days we hear a lot about this antibiotic resistance, right? So what are the bacteria prone to this resistance? And to understand these kind of problems, we apply metagenomics, and we get the information of the different microbiome profiles from the sequencing techniques. So then the advanced level of analysis is integrating all of these. So when if I have a sample, like different types of samples of same disorder, so then I would like to get a collective information or comprehensive picture of that disorder by integrating the analysis from genomics, transcriptomics, and so on.